Flowers are the mechanism by which plants reproduce. Each poppy flower produces thousands of seeds. The seeds fall into the soil where they germinate, grow and flower again. They often flower at the start of long dry spells, appearing to predict the weather. Farmers used to believe that picking poppies would actually cause thunderstorms. Leaving them would save their crops from the summer rains. External factors, especially temperature and light, are indeed important in the regulation of flowering. But other factors, including the plant's age and its genetic makeup, are important too. We'll look at some of the internal and external factors that regulate flowering, pinpointing some things that we do know and some that we don't. First of all, consider some effects of light that are well established. These chrysanthemums and many other plants sense day length. As autumn approaches, shortening days and lengthening nights trigger the whole population to bloom. So in some cases, the periodicity of light, or photoperiod, precisely regulates flowering. And one question that can be asked is where on the plant are the changes in day length detected? This question has been answered in experiments like this one with chrysanthemums, which are growing in the natural long days and short nights of summer. This is the control plant that will be exposed to natural conditions. On the plant next to it, the apical bud will be covered each afternoon at 4 o'clock and uncovered the following morning at 8 o'clock. So it's receiving a 16-hour night period and an 8-hour day. But only the apical bud, not the rest of the plant, which is experiencing natural long days and short nights. For the next plant, the leaves will be covered at these times so that they receive short days and long nights, while the apical bud will be exposed to control long day conditions. And finally, this last plant will have just one leaf covered each afternoon, leaving the rest of the plant in long day and short night conditions. This demonstration experiment will run in the 10 weeks it takes for the plants to flower over summer. It's being monitored by Ken Coxrell.
Well, the experiment's now been running 6 weeks, and some important differences are beginning to show up. This is the plant that we have kept in summer long day conditions, and as you can see, every growing point is still producing leaves. Now, this is the plant that every evening its apical bud was covered, and so the apical bud received a long night, while the rest of the plant was kept in summer long days. As you can see, that plant too is still producing leaves, and we can conclude from that that the apical bud is not able to detect the day length in which it is. Now, this plant is the one which every leaf was covered every evening and every leaf received a long night. And I hope it's apparent that there are flower buds appearing and developing quite rapidly on every growing point here. And we can conclude from that then that it is the leaves that are able to detect the long night. It is the leaves that produce a message, and that message is transmitted to the apical bud. When it arrives there, it instructs the apical bud to stop producing leaves, and to begin to initiate a flower bud.
This is a scanning electron micrograph of the meristem within the bud, and it's here that the signal from the leaves, which has been called phloragen, has its effect. Before that, the bud produces leaves. These are the first two leaf primordia, growing at each side of a newly formed apical dome. They show up as dark spots occupying about a third of the oval meristem. At this later stage, more leaf primordia have developed. The oldest primordium now has 3 lobes. Top left, there's a younger primordium, with another at the bottom. 2 more are just visible underneath. This is a typical vegetative or non-flowering apex. Leaf initiation has been modelled in 3 dimensions here. Each primordium will become a flattened leaf, and the process can continue for up to 30 leaves, with the apical dome gradually enlarging. If the enlarged apex now receives an appropriate signal from the leaves, flowering is initiated. The enlarged apex is surrounded by 4 primordia that will not develop into leaves, but will form the first of many scale-like bracts that surround the flower. Below them, here's the first glimpse of the flower primordia. These wrinkles are tiny florets or small flowers. Eventually, they cover the entire area of the apical dome. There are 2 types of floret in this chrysanthemum. The boundary between them is now showing. It runs right through the centre of the picture, separating them into central disc florets and outer ray florets. A fully developed flower, cut longitudinally, shows how the ray florets enlarge much more rapidly than those of the central disc. Returning to the experiment, flowers appear during the 10th week, but only on 2 of the plants, where all of the leaves were covered and where just a single leaf was covered. So, although the apical bud produces flowers, it only does so if the leaves, or indeed just one leaf, are exposed to long nights and short days. The induced leaves then send a chemical message to the apex that initiates flowering. We don't know what the message, or phloragen, is, or indeed whether several different messages might be involved. But we do know one substance that light acts on in the leaves. It's a molecule involved in the measurement of night length, which is the critical factor that triggers flowering. Plants are grown in green light to preserve this molecular trigger, and then it can be extracted. It appears as a faint blue pigment, a protein which is sensitive to certain wavelengths of light. It's called phytochrome. Phytochrome exists in 2 forms, and one is converted to the other immediately it absorbs red wavelengths of light, for example at sunrise. After sunset, this form of phytochrome slowly reverts back to the first form. This reversion process acts like a molecular clock that initiates the timing of the dark period. After a certain time, the flowering signal is generated. Like chrysanthemums, the flowering of poinsettia is regulated by photoperiod, and it also requires long nights and short days. So let's follow its growing cycle and see how phytochrome is involved in the onset of flowering. In summer, when the nights are short, day length comes far too soon and resets the clock. No flowering signal is generated because the grey form of phytochrome isn't present for long enough. But as summer turns to autumn and the nights get longer, resetting the clock is delayed. Eventually, the night's acts become long enough to generate the flowering signal. 12 hours around the September equinox for poinsettia, the leaves are now induced and flowering is initiated in the apical buds. This regulation of flowering by day and night length is called photoperiodism, and once flowering is initiated, nothing can stop it. The story recently became more complicated because it was discovered that phytochrome is not a single molecule. Phytochrome in the living tissues of another species, Pharbitis, can be detected using this special spectrophotometer, which measures the difference in light-absorbing characteristics of the two forms of phytochrome. That difference indicates when it's able to trigger flowering in the active form and when it's not, in the inactive form. In order to study green tissue of Pharbitis, where chlorophyll masks light absorption by phytochrome, a new assay was devised which uses antibodies raised against the phytochrome molecule. This immunoassay is 1,000 times more sensitive than the spectrophotometric assay. However, some of the phytochrome which was detected in the spectrophotometer couldn't be detected in the immunoassay. Brian Thomas.
The explanation seems to be that phytochrome is not a single molecule as we previously thought. It rather seems to be two molecules, which we call type 1 and type 2 phytochrome. These molecules each exist as an active and inactive form, but the molecules differ in their amino acid sequence. Finding these differences raises a whole set of new problems with regard to what phytochrome is doing in photoperiodism, and our current work is aimed at resolving what the roles of the different types of phytochrome are in this process.
Changes take place in the apex once the leaves are induced, that is, once they're exposed to appropriate daylings for flowering. These changes can be detected by feeding the plant with a particular form of the amino acid methionine, which becomes incorporated into proteins produced at the apex. In the same way, new proteins can be detected when leaves are induced. After extracting and separating them using gel electrophoresis, the newly formed label proteins can be located on autoradiographs. These experiments provide a snapshot of protein synthesis in the apex and in the leaves at different times after induction. It's been found that new proteins are being synthesized in the apex within 48 hours of the inductive treatment, long before any visible signs of change are apparent. The aim now is to identify the proteins involved and to determine the biochemical basis for their regulation. Brian Thomas again.
We have handles on the first and last steps in the control of flowering by day length. The real exciting challenge at present is to find out what happens in the leaf when it is induced. Induced and non-induced leaves to appearance look identical. However, we know there must be differences. We believe that there must be genes which are expressed in induced but not in non-induced leaves. These genes must lead to the expression of proteins which will be present in induced leaves and not present in non-induced leaves. We're trying to find these proteins. This is a difficult problem because there may be one or two changes in proteins taking place against a background of maybe tens of thousands of proteins in the cell. It's a bit like looking for a needle in a haystack. In order to undertake this search, we believe we're going to have to use the techniques of gene cloning and use these to search for particular messenger RNAs which are in induced but not in non-induced tissues. Screening for messages in these leaves is a large task. We believe we may have to screen something like 250,000 clones to find the messages that we're after. This is a considerable undertaking, but the rewards themselves are also potentially great. If we can find messages which are under day-length control, it will inevitably lead to new ways of manipulating flowering. In addition, we will have ways of manipulating other processes which are controlled by photoperiod. These could be processes such as dormancy and tuberization.
The discovery of the phytochrome timing mechanism can be applied to the commercial growing of chrysanthemums. This farm uses light to regulate the growing cycle so that the crops flower throughout the year and not just in their natural season. These cuttings are from the Canary Islands and Kenya, and they've been rooted by specialist propagators before arriving at the farm. They're irrigated through a series of micro-punched holes which run down the black lengths of polythene. This spray creates a fine mist which waters the growing crop. Heat is provided by the ribbed, thicker tube, which also runs the length of the beds. This heat circulates inside the tube at a temperature of around 40 degrees Celsius, and it supplements the overall heating of the greenhouse. So the environment of the crop is very precisely regulated. During the early stages, the crop receives cyclic night break lighting. The darkness of each night is broken up by regular short bursts of light, which are sufficient to reset the phytochrome clock to give short night conditions. A chrysanthemum crop growing under normal summer days with short nights will produce only leaves and no flowers. This suits the grower, who wants to have long stems on the plant and encourage the formation of leaves before the plant flowers. Remember that flowering requires long nights, 13 hours of continuous darkness, and this is easily done out of season by simply screening the crops. The cyclic night breaks are used by the grower to promote vegetative growth in young plants. By turning the lights on and off briefly for set periods, 2 weeks in the summer and 5 weeks in the winter, the plant is denied the long nights that initiate flowering and remains in the vegetative state until the grower is ready. The flower crop is also helped by enriching the growing atmosphere to 1,000 parts per million of carbon dioxide, more than double the normal concentration. As this gas is a byproduct of combusting natural gas, which is the main fuel used to heat the greenhouse, it represents no extra capital investment. A computer monitors this carbon dioxide enrichment, along with humidity and the temperature of the air and heating water. It automatically adjusts the interaction between these variables on the inside of the greenhouse to conditions on the outside. This minimizes the cost of producing flowers throughout the year. But for some crops, the problem is to prevent flowers being produced too early. David Hammond.
Celery is a herb of the umbelliferae, and it normally grows as a biennial, but as we grow it under glass with an autumn sowing and late winter planting for harvesting in May, it actually develops as an annual. What I mean by this is it will produce its seed stalk prematurely, and that can develop rapidly and form the problem of bolting, which of course makes the sticks that we sell on the market less attractive, and in fact in a bad season can cost the industry something of the order of £250,000 to £350,000 per annum on a turnover of about 5 million.
Bolting shows up first as a massive thickening and elongation of the base that makes the sticks inedible. How can this be prevented? Once again, scanning electron micrographs show the changes at the shoot apex that lead up to bolting. The apical diameter in celery, the distance across the apical dome between the two most recently formed primordia, increases with time and reaches a maximum usually in March. If flowering is induced, the apex then shrinks, indicating the onset of flowering. Beyond this stage, the apex switches from producing leaves to producing floral structures, and the unwanted flowers are the final stages of this development. At about this time, the stem height also begins to increase, slowly at first and then more rapidly. Beyond a certain height, the problem can't be cured. So part of the prediction of bolting involves regular measurements of apical diameter and stem height. These measurements also help establish which is the best cultivar to grow. This is Lauret, a cultivar which is not especially prone to bolting. A range of external regulating factors contribute to the success of this harvest. Prolonged exposure to low temperatures early in the growing season is one of the prime factors that can cause celery to bolt and flower. This process is called vernalization. If the crop is grown at a much warmer temperature than is currently practiced, then it will not be vernalized and flowering will be prevented or delayed. Although this costs money, it can reduce bolting. But celery also requires a particular night length to flower. So another tactic available to the grower is night break lighting. This has also been applied to constrain the bolt, and it's cost-effective, resulting in high-quality, marketable sticks. The problem of bolting is also being addressed in a completely different direction. These plants have been grown in daylit controlled environment cabinets that contain different concentrations of the hydrocarbon gas ethylene. The effects are dramatic. The stems of the plants are stunted and misshapen, and flowering is inhibited. By sampling the atmosphere in each cabinet, the amount of ethylene that causes these effects can be monitored throughout the experiment. This gas analyzer can detect the few parts per million of ethylene that cause stunting and inhibit flowering. For celery, it's no more than a scientific curiosity, but an effect of ethylene on the flowering of pineapple has long been recognized and used by commercial growers. They spray an ethylene-releasing compound onto growing crops in order to promote rather than inhibit flowering. The whole crop flowers and fruits synchronously instead of erratically, making it much easier to harvest. This is the last stage of development before the plants topple over and the fruit ripens. It would be very useful for growers if this kind of control were available for other crops. Knowledge of the external and internal factors that regulate flowering can therefore be applied. But despite more than 50 years of research, we still do not know precisely how flowering is controlled, and it remains an intriguing mystery.